Now we move on to the first part of quality control, that is internal quality control. Internal quality control is the study of errors and introduction of procedures to recognize and minimize them. The errors include all those arise within the laboratory, that is between the receipt of the specimen and dispatch of the laboratory reports. Internal quality control is managed by minimizing the pre-analytical, analytical and post-analytical errors. Any error in any one of these may lead to wrong test reports. In this slide you can see the common causes for pre-analytical, analytical and post-analytical areas. The slide shows how the sample is collected, that is patient preparation is done, then the sample is collected, then the test process is the procedure or processing of the sample is done. That is, if the sample has to be transported to another area or another place, the procedure is carried out. Then analytical means testing of the sample is done, and once the testing is done, then you move on to the reporting, report dispatching, preparation of the report dispatching and record keeping, maintaining the report is done. So in this slide it shows how the sample is collected and processed and reports are dispatched. So in every step of the quality control is maintained here. Coming on to the internal quality control variables. So monitoring of pre-analytical variables needs coordinated effort of many of the individuals and departments of the laboratories. Each individual must recognize the importance of their role in maintaining a high quality of services. So as already told, teamwork is very important here. To monitor and control these errors, it is necessary to understand each step involved in the laboratory investigation, and each of which can be reported, can be potential source of error. Coming on to the causes for pre-analytical variables. The first one is the test requests or ordering and utilization. Here the source of error may be due to inappropriate tests, bad handwriting or abbreviations used, and wrong patient identification. Patient preparation. Incorrect patient preparation can lead to errors. Laboratory tests are affected by many factors such as recent intake of food, medicines, alcohol, etc. by the patient. Smoking, exercise, stress, sleep and posture during collection of the specimen can also affect the laboratory tests. Hence, it is necessary to have the manual of instruction and procedure for patient preparation. It should be conveyed to the patients both orally and in written instruction, and phlebotomist must know these details very well. Next, specimen collection and patient identification. Various errors in collection can occur due to the following reasons. Selection of improper tube or anticoagulant selection. The container, selection of improper anticoagulant, that is if blood sugar is to be done, then you collect the blood sample in the EDTA instead of the going on to the blood sugar anticoagulated tube. Selection inadequate specimen. These include inadequate volume of the sample, excess anticoagulant, insufficient mixing of the specimen with the anticoagulant, inadequate type of sample. Example, hemolyzed specimen, lipemic specimen, or icteric sera. Icteric means jaundice specimen can also be seen. Specimen collected at wrong time. Example, if it is a lipid profile was asked, then it should be collected in a fasting blood samples. So these are the inadequate sample, mistakes seen under inadequate sample. Next, prolonged tourniquet application. By doing this procedure, what happens is local anoxia can take place. Anoxia means absence of oxygen in that particular area. That will lead to small solutes, leakage of small solutes such as potassium from the cell gets leaked, leading to increasing concentration of the potassium. Other reason may be causes excessive venous back pressure that leads to the concentration of the cells, proteins and substances bound to the proteins. Example, calcium. Blood collected from the arm into which administration of intravenous fluid is going on can also lead to dilution or contamination. Next, incorrect patient identification. The correct identification of patient specimen can be done by use of laser beam wands, that is which can read the barcodes. Next, mislabeling. Automated processing equipment or computerized specimen tracking system can eliminate mislabeling errors or lost specimens. When the specimen arrives in the laboratory, various logging and monitoring systems are necessary. Next, moving on to specimen transport. The stability of the specimen during transport from the patient to the laboratory should be monitored carefully. Specimens collected at the collection center and those sent to the specialized centers should be monitored more carefully. Specimen processing, the last. Inadequate spinning of sample tube may result in leakage of RBCs into the serum. This may result in contamination of the serum with potassium, inorganic phosphorus, LDH, etc. So these are the causes for pre-analytical variables. Coming on to the analytical variables. Analytical variables occur during actual testing of the specimens. To understand about this analytical variables, you should know the following terminologies. That is, first one is trueness. To know the trueness means it is defined as the closeness of agreement between the average value obtained from a large series of test results and an accepted reference value. That is trueness. Accuracy. It refers to the closeness, that is a degree of agreement of the estimated or measured value to that considered to be the true value. That is accuracy. Precision. Precision refers to the reproducibility of result, whether accurate or inaccurate. Inaccuracy of result may be due to improper standard or reagents, incorrect instrument calibration or poor technique. Precision can be controlled by replicate test, that is repeat test, check the tests or repeat the test of various measured specimens. In the slide you see how the accuracy and precisions are shown. In the first picture you see good accuracy as well as good precision. Where it is accurate, there only precision has been targeted. In the second picture you see good precision is there, but it is not accurate. In the third picture you see poor accuracy and poor precision is seen. Coming on to the analytical variables. We have finished with pre-analytical. Now moving on to the analytical variables. In analytical variables phase, errors may occur due to various reasons. These include selection of inaccurate method, mixing of specimen, use of instrument with poor precision, improperly calibrated instruments, improper volume of specimens, and presence of interfering substances in the test sample or specimen. The aim of validation of the test and calibration method is to demonstrate that the method is fit for desired purpose and that results have acceptable reliability. So that was variables of analytical procedures. What are the errors seen in the or source of errors for these analytical phases? The source of errors may be of two types, that is random errors and systemic errors. Random error, they may occur due to using wrong patient specimen or incorrect reconstituting the reagents. Other errors include pipetting error, transcription error, wrong numbering and labeling of the test samples, errors in calculation and errors in instrumentation. Systematic, the another error is systematic error. Systematic error may be due to wrong procedure, incorrect standard and incorrect standardization procedures. So we have finished with pre-analytical and analytical variables. Now coming on to the post-analytical variables. Post-analytical variable is mainly due to transcription errors. This can be avoided by verifying the results entered manually or through online instrument interference by the reporting or signatory analysts before the results are reported or dispatched. Sorry, I forgot to say about transcription. What do you mean by transcription error? Transcription error is nothing but mistake made when a person is performing data entry from one form of recorded documentation to another. That is transcription error. With this we finish with the pre-analytical, analytical and post-analytical variables. Next, moving on to the quality control charts. Before knowing about quality control charts, we must understand some certain terminologies. The first one being standard. Standard means this is the substance of constant composition of sufficient purity to be used for comparison purpose or standardization. Example includes glucose or cholesterol. Next, controls. This is a sample which is chemically and physically similar to known specimens.